the for dosing system. Before you are doing suspension, we have to do doping. Doping means should other biomaterial you have to do manually, but here it is doing automatically. It is a reading automatic scanning plate. Depends on which plate you are adding, it will automatically.
What do you mean by dosing?
Dosing is automatic dispensing.
Dispensing.
Yeah, that is the thing. Only that purpose only. It is the version is 8000. Okay. It is a touch screen and auto reading barcode. And this is the main switch. It is the here. You can see here.
Yeah.
Okay.
This side.
If you don't have any work, you switch off the. There is no need to always keep on.
Okay. Any maintenance also?
There is a maintenance is there every 6 months. For this one, there is no any maintenance. Only for the maintenance, we have to disinfect the this rack. Okay.
After switch on, there is no password or anything.
Nothing will also automatic. And this is the using for dosing racks, and it's especially for ID. You have to keep in here. This is the for normal. After you taking the colonies, you are using this one, inoculating this one, and you have to check the turbid. Okay, how much it is the turbid? That is the turbid is there. Each for TDR plate, each range will be there for turbid. Okay, according to that we have to inoculate. If you have more, you are adding the colonies, you should add with normal saline or pure distilled water. These two things. You have to keep always this one here, ID. Okay.
That is what solution?
This is the two solution will be there which you are using.
Susceptibility.
Yeah. This is the two. This one you have to keep your ID for
ID.
ID this one. It means
ID or ID?
ID. Yeah. Okay. Here. And diluent it is for fungal. It is not for other.
Yes.
And AST1 you always have to keep this position is AST1. It is the you can see the 10 ml. The bigger bottle is it is for 20 ml. It is not available here now right now. This is the things. If you want to add for fungal, you have to use for this one diluent. It will be there in the set all box. Okay. After preparing, you keep here, and this also open and you have to keep, and you have to use tips. You should not touch the tips. This is the tips. This is a 100 pack tips. You should use. I will take one tip for how to demonstration, and you use this one in the first position, not second position. Okay. Then, if you insert, is already is the plate is the look like a ELISA plate. Okay. Here is the well, and this plate has two portions. One is the ID for identification of the bacteria, and second was the susceptibility test. It is the antimicrobial susceptibility. This one. That depends on the indicators. It will be measures biochemical reaction, and same thing also here in normally 19 susceptibility test. It will be identified and it will be recognized which is the antibodies resistance or sensitive for the patients. Now how to open? This is the waste. We can remove the waste. This is all of the waste, and you should always careful in the biohazardical symbols. And when you are opening the running time, you should not open. It is the alert symbols. When you are loading the rack, you should be load like this position. ID should be backside, and you have to load like this. This is the for doping system. It is the AP100. This is the using. Either you can select manually. These all are the you are using the plate. E-cell paste I told you for the Strept and NH. And NF is non-fermentive bacteria.
As per this organism, we need to select the profile, right?
Yeah. No need to select. It will set automatically. It will read the barcode. It will identify. Okay. If you are using Strep, you have to use.
LAS connected?
No, it is not LIS
This is not
connected.
LIS.
LIS only is connected with this one. This is the for only doping system. Only automatically
No, we are
dispensing.
manually selecting according to this card.
No need to manual. You keep the card, it will automatically select. I will show you how to this one. Yeah.
No
Then
need.
only you have to give start.
Okay. No other purpose.
No other, no other purpose. Only dispensing. Because 96 main organism is inoculated here. From here, it will be dispensed to here. It will from 100 microliter dispense, and from here, it will dispense to everything. Okay. Now we'll turbidometer is there. More turbid organism will be terminated. It won't come proper report. According to the chart, you can see there the chart is there. You can see the green color chart. The TDR1 96, it is a 0.5 to 0.5. Before that, we have to, if anything is there, you keep first blank one without inoculating organism.
Could you try again?
You should keep this one, and if anything minus or plus one or coming, you have to make it as a 0. Okay, adjustment. You have to press here. Either like make it the 0, and it will get the 0. Then you have to inoculate and you check.
Just to show how to on it.
And you wipe this one always with the tissue paper, because moist also it will measure 0.1.
Coming now.
Yeah, you have to remove.
Should be 0.
It should be 0. Before you have to check, it should become 0. Yes. Should be 0.
Then only we have to start to measure the turbidity, right?
Then yeah,
Inoculate.
then you have to inoculate. Then you have to measure.
Yeah.
Okay. And you have to fit here. And make sure the plate cover should be open. You have cover will be there. You have to open and have to keep. Otherwise it won't dispense into the vial. It is for identification and susceptibility for testing. It has contained incubation chamber. It is 37 degrees centigrade, and we have 2 tower will be there. When it is blinking green, you can open and see A. It is a blank, and C. We have 32 tests we can load.
At a time.
Yeah, at a time 32 test or organism, whatever. And
What is the processing time?
it depends on it will take 24 hours. Some organism it will be take delay. It is depends on the incubation time of the organism. Okay. This is the you should after finishing you should close immediately. Okay. This is the main switch. Is no need to switch off nothing. You should go always on. Otherwise, your organism will be terminated. Okay. It is need for backup also for electricity.
UPS is required.
UPS is required for this one because it is an organism always going. Otherwise, that all not required. It is switch off also no problem. You can do. Now, main screen I will show you. Okay, can you see? Huh? Yeah. It is a two password you can use, an admin and user. And the user will be there, service user will be more access, and for admin is a limited access. And this is the low. You can come to the home. Sorry. Yeah. This is the main screen. This is the test, patient, result, QC, statistics, and nosocomial infection. Some infection, the details will be there in this one. You have to, according to the infection, you have to mention in the while you are processing. That time only it will be separate. Otherwise, no. How you have to go to here? This is the own set. This is the setting, this maintenance, and it help. You can search if any flag or anything, you can go for the help. It will be open, okay. And it is for the test processing sample ID. All you can go to here, and patient details you can find out here, and result also here. Okay. This is the own set. This is
ONET.
ONET, sorry. It is the analysis and data statistics for this one using. You can check the all data and the statistic from the ONET. Now we will go to the setting models. You can see common setup, select language is not necessary, but print setup and functional setup and data dictionary. You can open here anything. If you want to add to your report format, you can. We already added. If you want further, you can add. Okay. And functional setup, there is the date and time. It is the department, doctor, you can select this or which from blood, or we already default selection this one. If you want, you can in the report format you have to come, you can tick it to send diagnostic center, it will come to the report. And diagnosis and test, this already default this one, it will give the 10 to the power of 5 CFU. And this is the all antibiotics already is there. What are the antibiotics and sub-antibiotics, all we already default. Which are the you don't want, you can remove it. It depends on the doctor how they report. Here almost all of the microbiology in the worldwide, we are following CLSI guidelines. That's why
CLSI means?
that is the international guidelines for chemistry, all everything. We are, that is the CLS. that is Clinical Laboratories Society.
International.
So, that is just. And you have UQAST is there, European guidelines also if you want to follow. It depends on you, how you want to follow the antibiotic research. And clinical breakpoint. Some of the antibiotic don't have breakpoint. Some of the antibiotic have the breakpoint. This much, it is a strong. This much, less than it is shows like a resistance. It is showing more the sensitivity showing. It is the clinical breakpoints. It is the same like a normal range. It depends on the CLSI guidelines. We are following some other guidelines. If you change, it will be changes.
Criteria.
Yeah, it is like a criteria or protocol. This all coming to under to the CLS. Others under CLSI they not mention other like. You can check antibiotics. What are the CLSI guidelines or the breakpoints? So. And this is the only admin is there. If you want, you can create.
What about the QC?
Yeah, I will come back to it. Maintenance, there is a no daily maintenance. Only for cleaning the trays. Only that cleaning the any like trays cleaning with alcohol swabs or with sterile. You should not use any hypochlorite or nothing.
Alcohol swabs.
Only alcohol. Disinfectant. This system is something is like red. You can give to the system recovery. It will take 2 minutes to come. Okay. Yeah. Something it is like. You can check version or software version. You can check software version. This is not required. And help. You go to the help. Check to the help here. You go to search any flag, anything. You can check here. And now this is the same what instrument I showed here. A and C. There is a empty. See, there is a, if like this means column, there is a no plates. If any plates is there, it will shows the date also. There is a empty. 1632, 8 hour and 8 hour. You can load first plate also. Either you can enter the sample ID and again load also. There is a 2 option will be there. You can load, it will show the barcode, then you can go to adding the sample number. This is the quick apply, and you can here scan the barcode and you can keep it. If you press anything, you will get the all details. If your patient wise means this is the what are the red colors, it is a mandatory. You should fill this one. Okay. This is a very mandatory, and then this is already given. If you go to the here, you can see the patient information. You can see new patient information. You press new, give the all details, and you want to scan test. Here is the test code. There is the in the barcode of the plate. You have to scan it and you have to keep there.
So one by one we have to scan.
It will take automatically if you want to. If you want to select QC, here you can see here only you have to select QC and select the organism strains. which strains you want to use. This is a new one. This is the whole information. After that, we have to save. This is the save. After the information, you have to save. You want to delete, you can delete it here.
Okay.
Then, after that, everything you give the test barcode scan and you keep it here. It will take. It is like a manual type, like a other automatically. You have to go to the test here. You can view here, and it will show the plate here. You can go to here and you give the information on everything.
How we can run the test?
Yeah, I will
After
show you
keeping-
anything. Huh?
After keeping it, how we can run the test?
This is the thing. After keeping the sample, it will show us here. Okay. And you give the patient and you select here the plate already scanned here. It will come TDR1. Then you give the name, all everything, details. Then you save. Then you automatically it will recognize the plate. Shows here. You can search to here day to date wise. This one it is a up or minus or plus or minus, and you can see here breakpoints, lower and more or equal, and it is showing here. This is the report your resistance and sensitive.
MIC in the sense?
MIC is there is the values.
Full form?
I will tell you that I also forget MIC. This is all antibiotic you are getting the organisms.
Okay.
If you want to react, again you can give.
After 24 hours, same plate.
Same plate you can give, but don't remove the plate from there.
So then that you have an option, I didn't see that. Where is it?
Reanalyze.
Reanalyze, okay. Next day, report I.
There is a QC, and you can. This is the QC.
Patient.
All details you can see here. And
No.
reporting time.
Patient, this one is giving us QC. Or
This
any
is a
specific
QC. QC you have to give separate in the sense. You
QC
have,
material is there.
16 hours.
yeah, you have different strains and for a different organism.
Okay.
Okay. I
Okay.
think you have to give for Klebsiella is different strains and Staphylo is different strains. It's like 4 or 5 strains.
Okay. How we can run the QC?
We same way running. How we are taking first
Sample.
but QC material it is like a powder form. I think I guess we have to reconstruct and we have to streak
Daily we need to do.
No,
weekly once. It is a very expensive. If you
we
daily
have to keep back the QC in the field, sir. അതാണ് ഈ growth.
Okay.
ഈ growth ലാക്കിയിട്ട്
it is probability is more 91. Some other like 50-50 it will shows. Depends on the there is a positive or negative percentage. We have to identify the organism. Okay.
ക്യാപ്പ് പോയി കിട്ടും.
Cap is yeah.
D2 ആണോ D2 ആക്ച്വലി 2 ഓർഗൻസ് ഉണ്ട്.
You see, here is only one organism. If you sometime it will shows here, like a 50, 47 here.
Result equal to ഇതിന് സി വർക്ക് ചെയ്യുന്നില്ല
Yeah, area should become less than like a lower break one, higher break one, test result, report result. You have to test, this is a test, but the report result you have to give to the patient. And you will get the some messages.
Expert tools.
Yeah. Group U antibiotic are limited to the testing and reporting urine isolates. This to 4 doctors. Okay. According to that, they will choose the antibiotics. You can give in your report like a note. Either if you don't want, no need to do. Mainly important, you all this one have to concentration, MRSA, ESBL, MDR. These all are very important. Yeah. Vancomycin.
Yes.
Yes. And Clostridium. This all. The MDR. Report all. It is shows the in control. According to select. If you are wrong selecting ATCC strain, if it is done with the out of control, it is showing.
And our MIC very
For sensitive, one break for resistance. Always
actually break point should be like into 2.
equal to 0.5, less than 1, it is a sensitive. It's also sensitive. This is the monthly. As per as your lab, you are following how much test is comes over your QC, you will get the statistic. You can export also. You have to check. So you can change day to date. All you want, which are the either antibiotic side or organism or department wise or sample type. You can okay, then it will come here.
റിപ്പോർട്ടിൽ പോയിട്ട് ഡേറ്റ് ചേഞ്ച് ചെയ്യണം.
Here filter. Either start statistics.
ഫിൽറ്ററിൽ പോയിട്ട് ഡേറ്റ് ചേഞ്ച്
Here. Change. And then you filter. You can give here also. Doctor okay, special from the department. You can export or you can view.
ആൻറ്റിബയോട്ടിക് ആഡ് ചെയ്യുന്നത്
Yeah.
ഹൗ ടു ആഡ് ആൻറ്റിബയോട്ടിക്
That I told. What is your name?
Like just
Just full total or simply?
simply.
Simply, okay. This TDR X060 means TDR means only 6 test bottles we can load. It is the only for blood culture and it is from the venous or like arterial, either body fluid, either CSF. We have to inject to the, depends on the bottle. Two bottle is the aerobic or anaerobic bottles. Just it is easy to load. This is the main screen and load bottle and scan the bottle. Then open, it will blink, then you have to give. Yes, open and keep the bottle. Then after that we give okay.
Then it will go.
Yeah. Then this is the status. It will shows the which bottle you kept here. it shows the location.
Okay.
And growth curve we can see here.
Growth of the
Yeah.
microphone.
Algorithm graph we can see here when it is comes, which time is. The minimum detection time is 4 hours, 4.5 hours. Okay.
5 hours.
Yeah, means 5 hours. And this is the load. If you want to unload the bottle, you have to go to the software and you have to unload the bottle. Positive and unload the negative. Okay. And solid. It is some like a contaminator, like a other solar. Question mark it will come like that bottle. We have to remove from the here.
Okay.
And others. Reload. Lost probe. If any like alarm like symbol and the clean lost, you can press according to that. You can
Clear out all
clear
the
out of the lost the bottle. Okay. If you know or not.
that one.
Yes.